Hi, let's continue the introduction about the properties of biological molecules. So we have know about covalent bonds, and in this section, we will learn about non-covalent bonds. Non-covalent bonds are weaker than covalent bonds, but are crucial for biochemical processes, such as the formation of a double helix. Four fundamental covalent bond types are electrostatic interactions, hydrogen bonds, van der Waals interactions, and hydrophobic interactions. Ionic interactions, first we're going to introduce this. A charged group on one molecule can attract an oppositely charged group on the same or another molecule. It's called ionic interactions. The energy of an ionic interaction, sometimes called an electrostatic interaction, is given by Coulomb energy. Hydrogen bonds. These interactions are largely ionic interactions with partial charges on nearby atoms attracting one another. The hydrogen bond donor is the group that includes both the atom to which the hydrogen atom is more tightly linked and the hydrogen atom itself, whereas the hydrogen bond acceptor is the atom less tightly linked to the hydrogen. The electronegative atom to which the hydrogen atom is covalently bonded pulls electron density away from the electron atom, which thus develops a partial positive charge. For van der Waals interactions, the base of a van der Waals interaction is that the distribution of electronic charge around an atom fluctuates with time. At any instant, the charge distribution is not perfectly symmetric. This transient asymmetry in the electric charge about an atom acts through ionic interactions to induce a complementary asymmetries in the electron distribution within its neighboring atoms. The atom and its neighbors then attract one another. This attraction increases as two atoms come closer to each other until they are separated by the van der Waals contract distance. Some molecules, or terms nonpolar molecules, can't participate in hydrogen bonding or ionic interactions. When two such nonpolar molecules come together, some of the water molecules are released, allowing them to interact freely with bulk water. The aggregation of nonpolar groups in water leads to the release of water molecules, initially interacting with the nonpolar surface into bulk water. The release of water molecules into solution makes the aggregation of nonpolar groups favorable. The double helix is an expression of the rules of chemistry. So first, each phosphate group in a DNA strand carries a negative charge. Unfavorable ionic interactions take place when two strands of DNA come together. Thus, ionic interactions opposite the formation of the double helix. The strength of these repulsive ionic interactions is diminished by the high dielectric constant of water and the presence of ionic species such as sodiums or magnetic ions in solution. These positively charged species interact with the phosphate groups and partly neutralize their negative charges. And then hydrogen bonds. So hydrogen bonds are important in determining the formation of specific base pairs in the double helix. In single-stranded DNA, the hydrogen bond donors and the acceptors are exposed to solution and can form hydrogen bonds with water molecules. So when two single strands come together, these hydrogen bonds with water are broken and new hydrogen bonds between the bases are formed. the van der Waals interaction. So within a double helix, the base pairs are parallel and stacked nearly on top of one another. So the typical separation between the planes of adjacent base pairs is 3.4 angstrom, and the distance between the most closely approaching items are approximately 3.6 angstrom. So this separation distance corresponds nicely to the van der Waals contract distance. Bases tend to stack even in single-stranded DNA molecules. However, the base stacking and associated van der Waals interaction are nearly optimal in a double helix structure. So let's introduce the overview of genome information. And DNA is the carrier of genetic information in all cells and in many viruses. So DNA carries genetic information in the form of its sequence of nucleotides. The nucleotide sequence of DNA is transcribed into the nucleotide sequence of messenger RNA, which is then translated to a protein, a sequence of amino acids. The double-stranded or duplex nature of DNA facilitates its replication. So when a cell divides, each DNA strand acts as a template for the assembly of its complementary strand, and consequently, every pregnancy cell contains a complete DNA molecules. So genes direct protein synthesis. The link between DNA and enzymes is RNA. So the DNA of a gene is transcribed to produce an RNA molecules that is complementary to the DNA. The RNA sequence is then translated into the corresponding sequences of amino acids to form a protein. So as for transcription and translation, one strand of DNA directs the synthesis of messenger RNA, short for mRNA. The base sequence of the transcribed RNA is complementary to that of the DNA strand. The message is translated when transfer RNA, as we call the tRNA molecules, align with the mRNA by complementary base pairing between three nucleotide segments, so known as codons. Each tRNA carries a specific amino acid. So these amino acids are covalently joined to form a protein. Thus, the sequence of bases in DNA specifies the sequence of amino acids in a protein. The RNA that corresponds to a protein coding gene, we call it messenger RNA or mRNA, makes its way to a ribosome. As an organelle that is itself composed largely of RNA. It's the ribosome RNA, short for rRNA. At the ribosome, each set of these nucleotides in the mRNA pairs with three complementary nucleotides in a small RNA molecule called a transfer RNA or tRNA. Since the nucleotide sequence of the mRNA in turn reflects the sequence of nucleotides in the gene, DNA thus directs the synthesis of protein. So okay, that's all for today. Thank you guys. Bye.